Hi, everyone. Welcome back. Once again, I hope you and your families are doing well. And just as always, a reminder to read your emails, check out your announcements, and email me or see me on live chat if you have questions anytime. So today we are moving to chapter 20, which begins on page 117 of your course outline, page 413 in the text. And this chapter deals with biotechnology and genetic engineering, which is essentially manipulation of genes to have the outcomes you desire. And I saw this little comic strip when genetic engineering goes too far, and look at all these cows. This one gives whole milk, the next one 2%, one skim milk, and the last one with the coconut leaf on his head. Coconut milk, yay! Maybe they'll get that one of these days. Right now, that's not happening. But anyway, just looking at, first of all, when I teach this particular section, it's always challenging because there are so many tools and they all involve these different laboratory techniques. And especially when I'm doing this as far as distance learning goes and online, we're not going to be doing any of these procedures. So I'm going to do the best I can to explain them and show you some videos so you get an idea how they are done. But as far as the subject matter, I think it's really fascinating stuff. A lot of you have not been exposed to this. Even if you had a biology course, a lot of times they don't teach genetic engineering or DNA technology because there's so much new stuff going on. It's hard to keep up with. So I always try to rewrite this lecture every time and try to include some of the newest things that I can find. And keep in mind, it's always, always changing. Even the Human Genome Project that began in 1990, which was designed to identify all of the letters of our human genome of more than, we had that completed in only 13 years. This was a worldwide effort to do this, and they thought it would take decades, but because of high-speed computers, much of this is automated. It can be done much, much faster. And in fact, even with the coronavirus, they already sent out the sequence of that just, a few months or so after it first showed up. So it can be done much, much faster these days than we ever anticipated. So now we're working on sequencing the genomes of more than 7,000 species. And I wanted to show you just a part of an interview with Dr. Francis Collins. He was in charge of the Human Genome Project and what he has to say about it today.
The great enemy of writing isn't your own lack of talent, it's being interrupted by other.
Let me talk a little bit about something that you did that brought you to national fame and attention, and that's the Human Genome Project. What was the Human Genome Project, and why do we care about your having co-discovered it?
Well, good question. So what is the genome anyway? It's basically the entire instruction book, which is written in the language of DNA for an organism. So we humans have a genome, so do all other living organisms, animals, plants, bacteria, they all have a genome. Ours is pretty big. If you think of DNA as a language, it's an interesting one. It has just 4 letters in its alphabet, which we call A, C, G, and T because they're abbreviations for chemical bases. And our genomes are 6 billion of those letters. You get 3 billion from mom and 3 billion from dad. That's a lot, although it's pretty amazing to contemplate that that's a bounded set of information, and that seems to be sufficient to build a human being from a single cell. And when the Genome Project was started in 1990, we knew very little of that information. We had little snippets here and there of DNA information, but the whole thing loomed in front of us almost like an impossible task because our technology was nowhere near up to the ability to read that number of letters in any kind of measurable time.
So why is a person watching this or any human being better off because we now have mapped the human genome?
Well, there were many questions about that. The whole thing got finished in 2003, and there were some silly comments about, okay, medicine will be transformed in the next 2 weeks because of this. Of course not. 6 billion letters that you don't really know the language, it's going to take a while to figure it out. What's happened now over those last 14 years since then has, however, been transformative. in medicine and particularly in cancer. Cancer now, if you develop that terrible disease, you want to know exactly what misspellings have happened in the genome of those cancer cells that are causing those good cells to go bad. And that is a transforming capability that's now affordable because of the Genome Project and pretty much has changed everything in terms of the way in which we approach the diagnosis and treatment of cancer.
Today, I think it's the case you can have, anybody can have their human genome mapped for, is it $1,000 or less?
It's even a little less now, yeah.
And have you had yours?
You know, I have not had the whole thing read out, but I had a sampling of it, gosh, 8 years ago. I was writing a book about personalized medicine, and I wanted to use myself as a guinea pig. And so, yeah, I learned some things in that process of doing that about my own future risks that I found useful. One thing I did learn was that my risk of diabetes was substantially higher than the average person based on my genetic inheritance, and that was sort of a shock because that's not something that's run in my family. But my family are all pretty lean, athletic people, so maybe they managed to avoid it. That was at a point where I was not lean and athletic. I was kind of indulging in too many muffins and honey buns and not doing any exercise, and I was getting chunky. And that was enough to motivate me 8 years ago to change all of that, to lose 35 pounds, to get an exercise program. I'm a different person now than I would have been if I'd stayed on that same path.
Well, muffins and honey buns are not healthy for you? Is that what you're
They
saying?
are not healthy for anybody, I'm afraid.
Oh my gosh, it's like gone.
Okay, so you can see through that video that knowing these sequences has really changed a lot of things and how we approach them, including cancer treatments. We now can actually design personalized medicine based on what sequences you've inherited, what things have gone wrong, and so forth. So there's a lot to it. So when we look at this, there's a couple major goals that are usually involved in dealing with DNA technology and genetic engineering. And first of all, I just want to define the difference here. So when we talk about this term biotechnology, bio meaning life, right? This is the manipulation of organisms or their genetic components to make useful products. This has been going on for centuries as we do selective breeding programs in order to, breed cattle, for instance, that are heat tolerant and also can produce a lot of meat or something like that. And certainly we do it with plants all the time. Those making resistance and that sort of thing is more under genetic engineering, which is the direct manipulation of genes. And so we'll talk about the difference and some examples of all of these as we go along. But in general, what we're striving for is this idea of recombinant DNA. And recombinant DNA, that just means that you take DNA of two different sources and combine them. So they are recombinant. So it's a nucleotide sequences from two different sources. Often two species are combined in vitro, so in a laboratory setting, into the same DNA molecule. But in order to make recombinant DNA, you have to somehow get that DNA or that genetic material transferred from one cell to another. So here's a little bit about how we do genetic transfers.
Genetic transfer. Genetic material can be transferred between bacteria by several means, most often by conjugation, transformation, and transduction. Conjugation. Conjugation is mediated by a particular kind of circular DNA called a plasmid, which replicates independently of the chromosome. Many plasmids carry genes that confer resistance to antimicrobials. When two cells are in close proximity to each other, a hollow bridge-like structure, known as a pilus, forms between two cells. This allows a copy of the plasmid, as it is duplicated, to be transferred from one bacterium to another. This enables a susceptible bacteria to acquire resistance to a particular antimicrobial agent. Transformation. During this process, genes are transferred from one bacterium to another as naked DNA. When cells die and break apart, DNA can be released into the surrounding environment. Other bacteria in close proximity can scavenge this free-floating DNA and incorporate it into their own DNA. This DNA may contain advantageous genes, such as antimicrobial resistant genes, and benefit the recipient cell. Transduction. In this process, bacterial DNA is transferred from one bacterium to another inside a virus that infects bacteria. These viruses are called bacteriophages or phage. When a phage infects a bacterium, it essentially takes over the bacteria's genetic processes to produce more phage. During this process, bacterial DNA may inadvertently be incorporated into the new phage DNA. Upon bacterial death and lysis, or breaking apart, these new phage go on to infect other bacteria. This brings along genes from the previously infected bacterium.
Okay, so as you can see, there's several different ways that we can get DNA from one cell into another, or more specifically, from one species to another. And so often we use bacteria, and as that clip pointed out, bacteria, in addition to their single cell or their single circular DNA chromosome, in addition, they may or may not have these loops of DNA called plasmids. And usually these loops of DNA don't hold a lot of important genes that are needed for their own phenotypic needs, but instead um, they can, though, have one gene that's very important and it confers resistance, and that's what we call the R gene. And if they have that, we call that an R plasmid. So if there's a resistant gene, then many of you have probably heard about bacterial or that we have developed resistance to antibiotics. Antibiotics are pharmaceuticals and drugs that fight bacterial infections. And what will happen is if, let's say, you're given a course of 10 days on an antibiotic and you take the first, say, 5 days and you're feeling better, so someone else in the household is sick, so you give them the other 5 days, what you both have done essentially has you have taken the population that do not have this resistant gene out and left those that have a resistant gene. And then what they can do is pass that gene around through this process of conjugation. In conjugation, what happens is one bacterium will attach to another, as you see here, and they will pass a copy of that resistant gene over to their neighbors, so they can very quickly pass that around. So now you have a resurgence of these bacteria with resistant genes, so now you may get sick again, and the only way to combat that in the future is either take a higher dosage or take it for a longer amount of time. This is the same problem we have with pesticide resistance as well. So in the long run, this problem is never really solved. But here is how conjugation works again in more detail.
Conjugation is a mechanism of gene transfer that requires direct contact between donor and recipient cells. F+ cells carry the F plasmid, enabling it to make an F pilus and act as a genetic donor. Recipient cells that lack an F plasmid are referred to as F-. The F plasmid encodes for the F pilus, a protein appendage that attaches the donor to the recipient cell. The first step in plasmid transfer is contact between the donor and the recipient. The F pilus of the donor cell recognizes and binds to specific receptor sites on the cell wall of the recipient cell. The plasmid then becomes mobilized for transfer when a plasmid-encoded endonuclease cleaves one strand of the plasmid at a specific nucleotide sequence called the origin of transfer. A single strand of the F plasmid beginning at the origin of transfer enters the F- cell. A complementary strand to the single-stranded plasmid remaining in the donor is synthesized via the rolling circle mechanism. Once inside the recipient cell, a complementary strand to the single-stranded DNA is synthesized. When F plus and F minus cells are mixed together, eventually all of the cells become F plus.
So along that line of resistant bacteria, just a couple of words to think about. If you are given an antibiotic, make sure if you are prescribed an antibiotic that you take all your antibiotic. That way you bring that population low enough to combat your infection the first time, and then that would reduce the chance of breeding resistant bacteria. Also, you don't want to take antibiotics all too often because many times they will target their what we call broad spectrum kinds of drugs. So not only do they kill the infectious bacteria, but they kill good bacteria as well. And we have a natural flora that's really important, especially in our digestive system, that are often being killed off by overuse of antibiotics. That's why you hear so much about people taking probiotics if they're taking an antibiotic to try to hopefully keep their population of good bacteria up. And then finally, if you have a viral infection like very timely COVID-19, you don't antibiotics don't work on those. They have no antiviral properties. So that's why with viruses, a lot of times you're told just let it run its course. You can deal with the symptoms, but there's really no great way of. There are some antiviral drugs, and certainly we try to come out with certain ones for flus each year, but that's why they're scrambling so much with COVID-19 to try to come up with something. But it takes time, and the problem with viruses is they mutate quite quickly and frequently. So we'll talk about viruses here in just a minute. But back to this idea of conjugation and passing around these genes. So we start with whatever bacterium has the plasmid, we call that the F plus, that would be the donor cell, and then it forms that conjugation tube, and that word conjugation always refers to transferring some sort of DNA, nucleic acids. And so once they connect, then just like when DNA replicates, that plasmid unzips, it starts passing a copy or half of that strand over to the adjacent or neighboring recipient cell, which is called the F minus. And as that happens, that strand is being replicated. So the DNA replicates as it's being passed over to the F minus recipient cell, and now they are both F pluses. So again, this is all done through this conjugation bridge, which is called a pilus, is singular, or pili is plural. So how do we get DNA into cells again? So we can use conjugation where bacteria pass it around, or we can use something called transformation. And the term transformation is the word used for the taking up of DNA from the fluid surrounding a cell. And it's a natural um, occurrence. And so this happens in nature, especially with viruses and things like that. But if there's pieces of DNA and you mix those pieces of DNA with cells, they may or may not absorb it. That's essentially what it is. Once the DNA does get inside the cell, it has to be incorporated into the cell's DNA or chromosome by natural genetic recombination, or there's some things we can do to speed that up. But the point is, it's not considered a transformed cell until it actually takes up the DNA and it gets into its chromosome, and then it would be recombinant DNA. So then we would say the cell is transformed. So it will happen naturally, as it says here, or we can use something called electroporation, which is applying a brief electrical pulse to create temporary holes in the plasma membranes, and that will increase the chance that it will take up the genes that we're trying to get these cells to take up. Or we could actually use a gene gun, which is a direct injection of DNA into cells. We use this in plant cells a lot, not so much in bacteria, but it can be done. And this is showing this gene gun. It actually has plastic discs with DNA coated on gold particles, and it shoots just almost like a real gun. So we have our plant cells, and we take the particle gun, shoot them. The DNA gets into the plant cells, hopefully, and that those plant cells are transformed with that gene of interest, whatever that gene is. But here's how electroporation works.
Introduced into bacteria. Bacteria can then replicate and express that DNA.
Start that over.
Transformation is the process by which DNA is introduced into bacteria. Bacteria can then replicate and express that DNA, which can be isolated for downstream applications. Artificial transformation of bacteria involves producing competent cells capable of exogenous DNA uptake. There are two primary methods of artificial transformation used in the laboratory: chemical transformation and electroporation. It is thought that chemical transformation, which requires chemically competent cells, uses divalent cations to increase the permeability of the bacterium's cell wall, thereby increasing the likelihood of DNA acquisition. Many common protocols include a heat shock step to improve DNA uptake. Electroporation involves the application of an electrical field to the competent cells in suspension, thereby introducing holes in the cell's perimeter. The newly compromised cell wall allows exogenous DNA to pass into the cytosol of the bacterium. Outgrowth of bacteria following transformation allows for repair and recovery of the cells in liquid medium without antibiotic. After outgrowth, the culture is spread on selective plates to isolate transformants containing the newly acquired DNA, which conveys antibiotic resistance. A variety of competent E. coli types are available in different formats and for different applications, such as high-efficiency cloning and protein expression. Visit clonewithneb.com.
Okay, so once again, we're talking about recombinant DNA. How do we get DNA from two different species to combine? And in order to do that, we have to somehow manipulate the genes to get them to move from one cell to another. So we talked about conjugation, which is a gene transfer between bacteria using plasmids. Transformation, which is taking up of DNA from the fluid surrounding the cells. And again, in a laboratory setting, you could use a chemical method and heat shock to try to get that plasma membrane to become more permeable to the DNA, or you could use electroporation. And then if you wanted to do direct injection, you could use a gene gun. And there's other ways too, but these are just a few. And the third way is transduction, and that is the taking up of DNA using a bacteriophage, which is a virus that attacks bacteria. So a couple things about viruses. They're very species specific. Usually they don't cross species, but they sometimes do, as we've seen with COVID-19, which they think originated possibly from bats, and then there might have been even been an intermediate host. And so I want to talk a little bit about viruses here, especially because it's so timely with COVID-19 and going on right now, and tell you a little bit about how viruses reproduce, and then we'll talk about transduction and how we use viral vectors in order to actually inject uh, genes into whatever cells we're trying to infect. So we can use them to our advantage, but many times they are a disadvantage to us because they're infectious agents. So what is a virus, first of all? If you look at this bacteriophage, it looks almost like a little spaceship, and it's landed on the bacterial cell membrane, and you can see the nucleic acid being injected into the cell. And so what they are really are simply a protein coat and a nucleic acid. The nucleic acid can either be DNA or RNA. And if they're an RNA virus, they're called a retrovirus. We'll talk more about that, but that actually is what COVID-19 is. And so there's a little bit of advantage for us in combating an RNA virus versus a DNA virus and how they reproduce once they get inside our cells. But the point is that viruses are non-living. Because they cannot grow, reproduce, and metabolize on their own, they have to have a host cell to do that. We consider them non-living. And so they are infectious in that sense, in that they cannot live, they cannot survive, and they don't live actually because they're non-living, but they cannot survive or reproduce if they do not have a host cell. So when your immune system is fending off a viral attack, hopefully it gets the virus before it infects the cell, because once it gets into the cell, it takes over the host cell's reproductive machinery. And then in order to attenuate or stop the virus, you would have to actually kill the cell. And nobody wants to kill healthy cells, right? So here's a little bit about how viruses reproduce, and then I'll specifically talk about COVID-19.
Every cell in your body is a tiny factory, constantly making proteins. Your genetic code, or DNA, directs every activity. Protein-making machinery in the cell reads the cell's own DNA and makes sure the cell proteins go where they're needed. Viruses have their own genetic code, but they don't have the machinery to copy their DNA. So viruses need your cell's machinery to reproduce themselves. A virus starts this process by using the keys on its surface to gain entry. Like a Trojan horse, it enters the cell. The virus releases its own DNA into the unsuspecting cell and hijacks the protein-making machinery. Now, this vital cell function becomes an assembly line for new virus particles. No longer in service to your body, the cell now serves the virus, manufacturing thousands of new virus particles, which are released to enslave more cells, thousands and thousands more.
And so that's a really great video to show you how those viruses enter our cells, and it depends on those surface cell receptors. And that's why they're usually very species specific, because they have to match up with those receptors on those cells of the species they can infect. And so knowing that tells us a lot about the virus and how we can combat it as well. So first, this video on COVID-19, I'll talk about that, and then we'll talk about how we can use what we know about viruses in order to manipulate DNA in genetic engineering.
The large S glycoproteins are used by the virus to gain entry to human cells. They most likely attach to angiotensin-converting enzyme 2 receptors on the cell membrane, allowing the virus entry. The exact mechanism for this is not known. Most likely, as shown here, the human cell ingests the virus in a process known as endocytosis. Once inside the cytoplasm, the endosome opens to reveal the virus's genetic material, a single-stranded RNA. The virus hijacks the cell's machinery to replicate the RNA in N proteins and uses the endoplasmic reticulum to form its M protein outer layer and the all-important S protein. After replication, the virus is carried by the Golgi bodies out of the cell in a process known as exocytosis, so that it can infect other cells. Meanwhile, the stress of viral production on the endoplasmic reticulum eventually leads to apoptosis, or cell death. It should be noted that the mechanism of action for 2019 novel coronavirus is unknown. However, scientists in China have sequenced the genome, while others have used the sequencing to visualize the structure.
So again, the more we know about the structure of the virus, the more we know about how it can infect its host cell. And so, again, this third way of getting DNA into a cell, which is what viruses use, is called transduction. And it is the taking up of DNA. In this case, if we're trying to infect bacteria using a bacteriophage, and here is a generalized video of how transduction occurs.
In generalized transduction, a segment of DNA is carried from one bacterial cell to another by a bacterial virus called a bacteriophage or phage. The phage attaches to the bacterial cell and injects its nucleic acid into the host cell. A phage enzyme is produced that breaks down the host DNA into smaller fragments. Phage DNA is replicated and phage coat proteins are produced. During formation of the mature phage particles, a few phage heads may surround fragments of bacterial DNA instead of phage DNA. The phage particle carrying the bacterial DNA infects another cell, transferring the bacterial DNA to the new cell. When the bacterial DNA is introduced into the new host cell, it can become integrated into the bacterial chromosome, thereby transferring genes to the recipient. This cell then multiplies and carries new genetic material.
So you can see in that video clip, the phage actually injects DNA, as you see here, into the host cell. But in COVID-19, again, we don't fully understand the whole mode here of reproduction, but it seems more it's a process of endocytosis and exocytosis. So if you remember what that was, remember endocytosis where the cell actually engulfs it and brings it in, or endocytosis, or exo, that is, cytosis, after it's then reassembled all the new viral particles, it'll move out by exocytosis. So in that case, the cells aren't actually damaged. In some cases, the virus actually causes the whole cell to lyse, which would be rupture, in order to release all the new viral particles. And that's what happens in something like AIDS, where we see the helper T cell count goes way down because all those cells are actually being killed by the virus. So just a couple of words about COVID-19. This is from Johns Hopkins University. This is some of the best information I've seen out on it. And I just want to point out a few of these, but if you want to read all of these, I did send them out in an email, and I also, I believe, posted them under your announcements. But the first part is really important because this is kind of a pet peeve of mine. You hear people asking the doctors all the time, how long does the virus live on a surface and so forth? Remember, the virus is not a living organism, but an infective agent that typically consists of a nucleic acid molecule and a protein coat. Now, that's your typical virus, but COVID-19, couple specific things. It is a retrovirus, which means it's an RNA virus, and it also has an additional protective lipid layer. So remember, lipids are fats, and so when absorbed by the cells of the ocular, nasal, or buccal mucosa, which are also fat-based, that changes their genetic code and mutates them and converts them into aggressor and multiplier cells. So since the virus is not a living organism but a protein molecule, it is not killed but decays on its own. So it disintegrates. It doesn't die, it disintegrates, it falls apart. And that time depends on the temperature, humidity, and type of material where it lies. And on the next page, it gives you a lot more information about that. But keep in mind, the virus is very fragile. The only thing that protects it is that thin outer layer of fat. That is why any soap or detergent is the best remedy. It's anything that cuts the fat. So that's why you want to make sure you use a lot of soap, a lot of foam, wash everything, wash your knuckles, wash your thumbs. Don't forget anything. They recommend 20 seconds. And keep in mind also that heat melts fat. Therefore, it's good to use warm water when you wash your hands, your clothes, and everything else. And then it goes on to talk about all these other things. But this point here, no bactericide serves. The virus is not a living organism like bacteria. So antibiotics, just like when you take oral antibiotics, if you're using antibacterial soap, it's not necessarily the antibacterial aspects aren't going to affect the virus, but the soap will, because the virus has that uh, fat layer on it that will be broken down by soap. So again, you can read through this. There's more information here, and again, it went out in an email, and you can feel free to pass that on. Okay, so back to DNA technology and looking at concept 20.1, now on page 117 of the course outline, and how, first of all, we clone DNA. This is just kind of a preview of making recombinant DNA and then cloning it. And I think everyone knows by now cloning means making exact copies. So essentially what we do in genetic engineering is we try to get a gene of interest, whatever our gene is, and let's say it's the gene for insulin production, which I think you guys know is a hormone that helps regulate blood sugar. And people who are diabetic need to take insulin. And there was a time when we had to actually kill animals and use their pancreas in order to get insulin and use it as a pharmaceutical. Now, fortunately, we can use genetic engineering to cut our gene for that codes for insulin out of one of the human cells. So here's the cell that contains the gene of interest. Let's say it's the insulin gene. And we want to then insert it into a plasmid and then get the bacteria to take up the plasmids that have our gene of interest so that they will produce the proteins. Remember, bacteria go through the same stages we do, transcription and translation, in order to make proteins just like we do. They're faster though, because remember, they're prokaryotic cells and they don't have a nucleus and everything is happening simultaneously and it's not as complicated and they don't have as much DNA, so they can do it much faster. And that's why they reproduce so much quicker, and that's why we use really bacteria as little protein factories. So first we need to get the gene inserted into a plasmid using one of the methods that I mentioned, conjugation, transformation, or transduction, and then we need to get that plasmid put into a bacterial cell. And these days, actually, you can order plasmids that have the gene of interest that are in what we call a DNA library. They just keep them in a collection, keep growing the bacteria that have them, and you can get those from biological supply houses. So number 3 here, the host cell is grown in a culture to form a clone of cells. So remember, when bacteria reproduce, they reproduce very much like mitosis. It's called binary fission, and they make exact copies. And so that's what cloning is. And then the protein expressed from that gene of interest is harvested, and you can use it, as I said, as a pharmaceutical. This is how we genetically engineer insulin, also human growth hormone that treats stunted growth. We can use this protein to dissolve blood clots in heart attack therapy, for example, or we can take the actual gene of interest for basic research on genetic engineering of, say, genetically engineering plants for pesticide resistance. So we hear a lot about GMOs these days. I'm going to talk a little bit more about those later. If you're pro GMO or no GMO, we'll help you look at the details of that. And then also, this is a great one. We were able to use the gene to alter bacteria for cleaning up toxic waste. And so oil spills were a huge problem, but we actually have genetically engineered bacteria that can break that down. So some really great outcomes here. So now looking at using restriction enzymes to make recombinant DNA. So these are going to be enzymes that are going to be our DNA scissors. So keep in mind what we're trying to do here when we design recombinant DNA is we have to cut the DNA out of one source. So we need DNA scissors, and then we're going to paste it in to another chromosome. So we need DNA paste. We've talked about a lot of these enzymes already when we looked at DNA replication and protein synthesis. And so we use what we already know happens in nature, bring it into a laboratory setting, and get it to work here in order to create bacteria to produce the kinds of proteins we need. So bacterial restriction enzymes are the DNA scissors. They cut DNA molecules at specific sequences, and those are called restriction sites. So a restriction enzyme makes cuts at specific sites, and depending on what you're trying to do, you can actually order these enzymes to cut after certain sequences. Like this one enzyme will always cut after AATT or something like that. The most useful restriction enzymes cut DNA in a staggered way that produce fragments called sticky ends. And then what happens is they will complementary base pair and will seal that with DNA ligase. Remember, ligase was associated with the discontinuous side of when DNA was replicating, and it filled in those gaps. And I told you back then that that is our DNA paste. So here we see ligase coming into play again, which is going to be the DNA paste. So restriction enzymes are the scissors, ligase is the paste, and here's how that works.
Restriction endonucleases are enzymes that cleave DNA at specific nucleotide sequences. The sequence recognized is often 4 to 6 nucleotides long. For example, the restriction endonuclease EcoR1 recognizes the sequence GAATTC. The nucleotides at one end of the recognition sequence are often complementary to those at the other end. The two strands of the DNA duplex have the same nucleotide sequence running in opposite directions for the length of the recognition sequence. Because the same recognition sequence occurs in both strands of the DNA duplex, the restriction endonuclease can bind to and cleave both strands of the DNA molecule. Because the bond cleaved is typically not positioned in the center of the recognition sequence, and the DNA strands are antiparallel, the cut sites are offset from each other. After cleavage, each DNA fragment has a single-stranded end a few nucleotides long, and the single-stranded ends of the two fragments are complementary to each other. These single-stranded ends can pair with each other, sticky ends. Once their ends have paired, the two fragments can be joined together with the enzyme DNA ligase, which reforms the phosphodiester bonds of DNA. What makes restriction endonucleases so valuable is that any two fragments of DNA produced by the same restriction endonuclease can be joined together. Restriction endonucleases are fundamental tools in genetic engineering.
Okay, so a closer look at how that works. Again, we start with restriction enzymes that are going to cut at a specific restriction site. And again, this one happens to cut after the TTAA. And so we use that enzyme to cut this particular DNA. And we also use that same enzyme to cut the DNA of the other source. Remember, we're trying to put DNA of two different sources together to make recombinant DNA. And so the DNA fragment added from the other molecule cut by the same enzyme then will base pair. So these two T's bring up the two A's bring up the two T's. And so here's a combination. And then, of course, we need our DNA glue, which is DNA ligase, to seal the strand. And now you have your recombinant DNA, that is DNA from two different sources. So once we get these plasmids, we want to clone them in order to keep copies of them. As I said, once you get this, you can patent it, clone it, sell it, be a millionaire, good stuff. In gene cloning, the original plasmid is called the cloning vector. A cloning vector is a DNA molecule that can carry foreign DNA into a host cell and replicate there. And so there are several steps required to get to this point. And first I'll show you this clip on the steps in genetic engineering, and then we'll go through each step.
To fly a rocket ship, you need to be an optimist. No astronaut launched.
Bacteria have been helpful to genetic engineers, not only in providing them with restriction enzymes, but also with what are called plasmids. In addition to their single chromosome, many bacteria also contain tiny rings of DNA called plasmids. Plasmids are usually about 1,000 to 100,000 nucleotides long and act as independent, self-replicating molecular operators within bacteria. Plasmids are one of the ways that DNA recombination goes on within nature. While a bacteria's chromosome contains all the necessary genes to code for the bacteria's existence, plasmids provide an effective way by which traits not contained in the chromosome can be passed from bacteria to bacteria. For example, some plasmids contain genes that code for enzymes that digest certain antibiotics, such as penicillin or ampicillin. This is obviously an advantage to the bacteria. When a bacteria containing these plasmids dies, it breaks open and liberates these plasmids to the outside environment, and they are often taken up by other bacteria that then acquire the traits coded for by the plasmids. Genetic engineers can place liberated plasmids in a solution with restriction enzymes and cut them apart. The engineers then place the gene segment they wish to insert into the solution with the plasmids, and then add repair enzymes that join the pieces of DNA together at their sticky ends. These new plasmids, with their newly inserted genes, are then placed with bacteria into a different solution that enables the plasmids to readily penetrate through the cell wall and membrane of bacteria. The bacteria that are exposed to the plasmids are taken out and grown in a culture for a short time. If the plasmids into which the experimental gene, such as the gene for the production of insulin or growth hormone, is inserted, also have genes that provide resistance to ampicillin and amoxicillin, a unique technique can be used. Genetic engineers know that not all the bacteria exposed to the plasmids will absorb them. By exposing the cultured bacteria to antibiotics such as ampicillin and amoxicillin, they kill off the bacteria that didn't absorb the new plasmids, while those bacteria that did absorb the new plasmids containing the genes for antibiotic resistance and for, say, insulin or growth hormone continue to grow and thrive in the culture. Scientists continue to grow these bacteria until the insulin or growth hormone produced by them is sufficient to be extracted and purified for use by human patients. But how are genes inserted into the chromosomes and DNA of more complex organisms such as plants or animals? There are two major methods of placing DNA into the nuclei of eukaryotic cells: vectorless and vector methods. Vectorless methods of insertion of DNA don't require the use of a third intermediate organism to deliver the DNA from one species to another. Vectors, in the biological sense of the word, are organisms that carry the DNA of one species and insert it into another.
Okay, so if you look at your outline on page 118, you will see the steps involved in a typical genetic engineering experiment. And I have them broken down into stages, and I'm going to go through each stage with you here, beginning with stage 1, where again, the idea is we're making recombinant DNA. So we begin with DNA from two sources, and I'm going to stick with the same example I was using. Let's say the DNA from the animal cell is going to be the insulin gene, and we're going to use E. coli as our bacterial factory, which again, this is Escherichia coli, which is a common inhabitant of our digestive system. So it's a very common bacterium. And we start out then, number one, the DNA from two sources is isolated and cleaved with the same restriction endonuclease, right? These are our restriction enzymes, also known as endonucleases, which are DNA scissors. So we cut them with the same scissors so that we get the same restriction sites. And then stage 2, the two types of DNA compare at their sticky ends. So the sticky ends, remember, when we have these staggered cuts, and when they're mixed together, DNA ligase is going to join those segments. Now, here's the thing about the plasmid we're using from E. coli. It's got two important genes of interest. It has this LAC-Z gene, which is a gene to help metabolize lactose, and when it does that, it will turn blue on an agar plate. So you guys know what an agar plate is, right? The plate that had gelatin in that we use for diffusion. Well, we can lace it with particular kinds of substances like lactose so we can identify these, because we really have to do some detective work in order to figure out which bacteria has taken up the gene of interest. So we use this LAC-Z gene, and we also use an ampicillin resistant gene. So what's ampicillin? Hopefully you know that is an antibiotic. And it is an R gene. I mentioned the resistant gene. So if we try to grow bacteria on ampicillin-laced agar and it doesn't grow, we know it has this gene. If we grow it on lactose and it metabolizes it, we know that this LAC-Z gene is functioning. So this is going to help us with our detective work later. So once we get the recombinant DNA, now notice all these plasmids, we don't know which ones are taking up DNA and which ones are more specifically taking up the gene of interest. This one didn't take up anything. It's not recombinant at all. It's just like this one. This one took up some DNA which broke up the LAC-Z gene, which means it can't metabolize lactose, but it's the wrong gene. We're looking for this gene. The gene of interest is red. This one also has a non-functional LAC-Z gene because it's got recombinant DNA, and that broke up the gene, so it won't work, but it's not the right one. We're looking for this one. So how do we find it? So the first thing we do is we take all the plasmids and we hopefully transform the bacteria so they take them up. And then in stage 3, after these plasmids are inserted into the bacterial cells by transformation, we let the cells reproduce and form clones. So now we have all these clones, and now on to stage 4 where we're going to screen for the gene of interest. How are we going to find which one's which? Because this batch is recombinant, but it doesn't have our gene of interest. This batch is recombinant, but doesn't have our gene of interest. This one's not even recombinant. This is the batch we're looking for with the red gene. Hopefully you guys are with me on this. Okay, on to stage 4. Now, stage 4 has 2 different parts to it, the preliminary screening and then the secondary screening. In the preliminary screening, the bacteria are plated on a type of agar that selects for the bacteria with recombinant plasmids. That is, anything that has recombinant DNA will not be able to break down this lactose agar, right? So the blue shows the colonies with the plasmid. Sorry, this is ampicillin. I got ahead of myself over here. So let's start with this. First, we're going to eliminate the cells without the plasmids by growing them on ampicillin-laced media. So all the plasmids had a resistant gene. And so any cells that don't have the resistant gene will not be able to survive. So the cells that did not take up the plasmid are not resistant to ampicillin and do not form colonies on media containing this antibiotic. By the way, what is a colony? When bacteria grow, they grow in these little clumps of all the same type called colonies. So these are the colonies then that were able to survive. What we did essentially is we killed off those that didn't have the resistant gene, and now we know all of these have a resistant gene, which means they also all have plasmids. So the next thing we want to know is which cells, or identify the cells without recombinant DNA. And so we know the ones that have recombinant DNA have had their LAC-Z gene interrupted and cannot metabolize this media. So any of the ones that don't turn blue are the colonies with recombinant DNA. Those that do turn blue, then they do not have recombinant DNA because their LAC-Z gene is still functioning. So again, to conclude, cells that did not take up DNA fragments have a functional LAC-Z gene and are able to metabolize the xgal and turn blue. So now we know which cells actually have recombinant DNA. But we don't know, out of those that have recombinant DNA, which one has this gene. We need to find the specific gene. We know they all have some gene. We don't know which ones have the gene of interest. And this is where stage 4, our secondary screening, comes in. And here we're going to use radioactively labeled probes to identify the gene of interest. And this process is called nucleic acid hybridization. So for example, if this is the code for the desired gene, then we would synthesize a probe that is complementary to it. So if this gene codes for insulin, the complement to that, C would be G, T would be A, C would be G, and so forth. And then what we would do in this last stage is, first of all, the colonies of the plasmid containing bacteria, each from a clone from the clone library, are going to be grown in some agar. This is the gelatin. And then number two, what we want to do next is make a replicate of the plate using filter paper. So we just press this filter paper against the colonies, and some cells from each colony then adhere to the filter paper. The reason we want to do that is because we're going to, unfortunately, sacrifice these bacterial cells as we experiment to figure out which one has a gene of interest, while we keep some of the cells behind growing in the plate. So the next thing we do is a series of procedures that will denature the DNA, break it open, that's why it kills the cell, and then look for that particular gene of interest using that radioactively labeled probe. The probe contains the nucleotide sequences complementary to the gene of interest, and it will bind to cells containing that gene. So number 4, only those colonies containing the gene will retain the probe, emit radioactivity, and we can see it on this filter paper. So now what we do is we compare the original plate to this filter paper, go back, and there we have our gold mine. Yay, that's the one that has the gene of interest. And so now we just keep that, clone that, keep it in a genetic library, and we can sell it and make millions, maybe. Depends on what gene it is, right? So what is a genomic library? Just like a regular library is a collection of books, in this case, it's a collection of genes. So it's made using bacteria that the collection of recombinant vector clones produced by cloning the DNA fragments. And we also have a new term here, complementary DNA. So this is abbreviated cDNA. And what this is a library that is made by cloning DNA made in vitro by reverse transcription. So what is that? Well, let's think about this. Transcription is converting what to what, right? Transcription is converting DNA to RNA. So reverse transcription must be converting RNA back to DNA. Now, why would we want to do that? We want to do that because, remember, bacteria don't have any non-coding regions. They cannot read any of our introns. So we have to have an intron-free copy of DNA in order to put it into bacterial cells. And the only way we can do that is to take our RNA after it's been processed and convert it back to DNA. And that's where this special enzyme called reverse transcriptase reverses transcription. By the way, side note, I mentioned that COVID-19 is a retrovirus, and that is, it's an RNA virus. Well, in order for it to infect our cells, it has to convert its RNA to DNA. That's where we got this particular enzyme known as reverse transcriptase. It's a viral enzyme, and it's only found in RNA viruses, and they have to convert their RNA to DNA to continue on with their takeover of our host cell machinery. So if there's one of the things we've used, and you may have heard that they're looking into some of the HIV drugs to help with this COVID-19, is because HIV is also a retrovirus, and we have reverse transcriptase inhibitors. That's part of that cocktail that originally came out, which inhibits reverse transcription from taking place, which then interferes with the virus. So that's one way we can attack a virus without affecting our own cells, by interfering with their reverse transcription. That enzyme is unique to those viruses. So it's a little bit of an advantage that COVID-19 is a retrovirus, and that maybe we can use reverse transcriptase inhibitors to help slow it down. It won't get rid of it, but it will slow down the reproduction. So anyway, back to this idea of cDNA. So cDNA is made from using this enzyme, reverse transcriptase, and it then produces an intron-free piece of DNA. So here's how we make cDNA. So we start with our regular DNA, let it go through transcription so that we have, and all the way through transcript modification until we end up with our mature messenger RNA transcripts. And then we just do what we know happens in nature, only we do this in a laboratory setting, is we take our intron-free messenger RNA using reverse transcriptase, there's that enzyme, we can convert it to DNA. And then what happens is we break down that messenger RNA transcript, and now we have our intron-free copy of DNA, and we just make copies of that. So how do we do that? Well, we use DNA polymerase. Just like when our DNA replicates in nature, we do this in a laboratory setting. And so in order to do this and make copies of this DNA, once we get this DNA, we want to make many, many copies of it. We can amplify it in vitro using something called the polymerase chain reaction, or PCR. This is on the bottom of page 118. This was a huge breakthrough because before polymerase chain reaction or PCR was available, we just had to keep growing many, many, many hundreds and thousands and millions of tiny cells in order to make copies of that cDNA. But now we can just put it in a machine, and overnight we can have hundreds and thousands of copies. So it's all automated, it's all done by computers, and it's a 3-step cycle that involves heating the DNA, cooling it, and then letting it replicate. So we do need to have DNA polymerase as our source to bring up the complementary bases. But the problem with regular DNA polymerase is that it will break down if we heat it, just like all proteins and our nucleic acids will denature if we heat them. So we were fortunate enough to find heat-sensitive enzymes, or heat-resistant enzymes, that is, in the ocean's thermal vents. We found bacteria that have enzymes that can handle that kind of temperature. And so we were able to use this unusual heat-stable DNA polymerase called Taq polymerase. And it's got this particular sequence, GTCA. And so you'll hear Taq polymerase. That is used then in PCR. So we can actually do this. We have on our campus a PCR machine that can make copies of that DNA. So here's how it works. We start with our target sequence. And by the way, this can not only be copy DNA that we want to make copies, but this is very often done in crime scene investigations where you get just a little bit of DNA, say, at a crime scene, and the first thing you want to do is extract that DNA from the cells and then make copies of it so that you can do DNA fingerprinting and you have a lot of material to work with. This is one of the problems with the earliest use of DNA fingerprints, is that you didn't have a lot of chances to redo it if something went wrong. So sometimes it didn't hold up in court because you couldn't be sure there wasn't some kind of infectious or problems in the lab that caused it to show what the results it did. So here you can prove it because there's so much DNA, you can do it again and again. And so now our DNA fingerprints are very foolproof, really. So we start with that target sequence of DNA, and as I said, it's a 3-process situation. We start by denaturing, that's going to cause it to split. And annealing is our second step, which involves using those little RNA primers to begin with that tracks the, in this case, Taq polymerase. And we start making copies of either side, and that is extension. Once we get the 2 copies made, then those 2 copies go on to make 4, and those 4 to make 8, and it continues exponentially. So as I said, very, very quickly, overnight, you can have many copies of this DNA for whatever purposes you need. So our third cycle here, as you can see, yields 8 molecules. So a couple things I wanted to share with you. So you can actually see how this machine looks, this PCR song. It's actually an advertisement for this particular PCR, but it's really pretty hysterical. And as you listen to it, see if you can follow the words, and it will explain PCR and why we use it.
There was a time when to amplify DNA,
you had to grow tons and tons of tiny cells.
Ooh, and along came a guy named Dr. Carey Morris.
Said you can amplify in vitro just as well.
Just mix your tablet with a buffer and some primers, nucleotides and polymerases too.
denaturing, annealing, and extending. Well, it's amazing what heating and cooling and heating will do. PCR.
All the scientists out there doing PCR, Biorad salutes you with the all-new 1000 series thermal cycling platform.
Okay, so I hope you got a kick out of that. Maybe you even recognize that song. Depends. It's an old one, but it is a spoof on We Are the World by Michael Jackson. He had a concert to some sort of aid concert way back when, and that's what that song is all about. Pretty funny. But hopefully you were able to follow those words, and my question to you is then, what can you use PCR for? So usually when I ask this in a class, everyone says to find out who's your daddy because everyone could hear that part pretty well. And that's true. It's used in paternity suits to absolutely prove paternity. Remember, blood types can disprove paternity, but they can't actually prove it. So yes, it's used for that. It's used to solve a crime. As I mentioned, crime scene investigations, we use it all the time. And also, as I said, to make that copy DNA or cDNA, complementary DNA that is. So they made a sequel to this, and it's about Taq polymerase, which again is pretty funny. So I'm going to go ahead and play this. We all need a laugh at this point, right?
Six years of graduate school, and look at our boy now. Oh, Randolph. Now, I remember somebody that wasn't too different from this not too long ago.
Enzyme, what you doing today? I said enzyme, want to make DNA? I said enzyme, show me your binding way. Let me mix you with some primers. Enzyme, time to build a new strand. I said enzyme, cause I know that you can. I said enzyme, these letters also spelled and all we need to get things started.
To all the scientists out there doing PCR, Biorad salutes you with the all-new line of SoFast Super Mixes for real-time PCR.
Oh my gosh, I never stop laughing watching those. That guy is really good, isn't he? He's got a good voice and good rhythm. The whole thing cracks me up. But again, unless you're a science nerd, I don't know whether you're laughing with me or maybe laughing at me. So that brings us to why do we use PCR? One of the reasons we use PCR is so that we can run DNA fingerprints. So how do we do that? So now we're moving to page 119 in the course outline, looking at gel electrophoresis. And one of the things we actually do in lab, one of these techniques that we do in real lab if we were face to face, is to run a DNA fingerprint, and we would actually extract some DNA, run a fingerprint, and do all of this. But since we are now online, we're just going to do, I gave you a virtual lab to do for this, which is, but you can actually extract DNA at home if you want. You won't be able to run it unless you have a gel electrophoresis at home, but you can at least extract DNA if you can get your hands on some strawberries, I think it is. Anyway, it's not that hard, but just keep in mind, let's start with a look at a crime scene investigation. So we go out to the investigation. We usually can get a little bit, maybe cells somewhere. Most cells will have DNA, with the exception of blood cells lose it pretty quickly. They lose their nucleus. So, but as long as you can get some kind of DNA, then the first thing you would do is take it back to the lab and extract it from the cell. And then you would amplify it using PCR, make extra copies, and then you could run it through this gel electrophoresis machine. And so this technique then uses a gel as a molecular sieve to separate nucleic acids or proteins by two things, size and electrical charge. And it's a lot like we saw when we were using our agar plates, and we saw that larger molecules diffuse slower than the smaller molecules. So size will influence the rate that they will diffuse through this gel, and then electrical charge will move them through the gel. So we'll put an electrical charge through the gel because DNA is negatively charged. It's got negative phosphate groups, and it will be attracted then to the positive end. And then we're going to see variations depending on that size, and that will then be the fingerprint that we're looking for. The sequence changes that alter restriction sites are called RIFLEPs for short, restriction fragment length polymorphisms. And so polymorphism, poly meaning many, morph means shape. There's many shapes because when we take that DNA, we want to first, after we extract it, we want to cut it up with restriction enzymes, different ones, so we get different cuts. And the more we use, the more different cuts we have, the more precise the fingerprint would be. So this is how it works. We take our mixture of DNA, let's say the DNA from a crime scene. Let's say we have some suspect's DNA, we have crime scene DNA, and or you could have, parents' DNA in a child, whatever it is. And you take it and you inoculate these wells, little cutouts in the gel, and notice that there's a negative cathode and a positive anode. And when we turn this power source on, it's going to attract those negative phosphates in the DNA towards the positive anode. And also the size is going to determine that pattern. So the shorter molecules diffuse faster. So they'll be down at the end of the gel where the longer molecules will hang back. And here is your pattern then. Now you can do a couple different techniques to view this fingerprint. You can, and just a word about the DNA that's used for the fingerprint, by the way. You have to be careful to use DNA that we don't all commonly share. So you don't want to pick DNA, say, that would code for some similar traits like, I don't know, blood type or whatever. You would want to use instead DNA that's in those nonsense regions. And so we have these repetitive sequences of DNA that are really non-coding, and those are that would be the best DNA to get. And so it's kind of like the DNA that codes for your real fingerprints. There's no advantage or disadvantage to having that DNA, so it's very randomly dispersed in populations. That's why my real fingerprint is very different from yours. So it's kind of the same idea. But anyway, here's how the fingerprinting works.
My house was broken into last week. The police can identify the culprit from blood found at the scene. How can they do that? To understand this, we need to state what is meant by DNA fingerprinting and describe some uses of DNA fingerprinting. The burglar smashed a window, leaving blood on the broken glass. Samples of blood, hair or skin can be collected from a crime scene. Forensic scientists investigate the samples in a lab. First, they extract DNA from the cells. They use enzymes to cut the DNA into shorter fragments. The fragments are placed into a well in a gel. They are separated using an electric current. Different sized fragments travel different distances in the gel. The smallest fragments travel the furthest. A pattern of bands is produced. This can be compared with the patterns of DNA from possible suspects. The enzymes cut different people's DNA in different places, producing a different set of fragments. The different sets of DNA fragments will produce different patterns of bands when separated on a gel. Each person has unique DNA, which will produce a unique pattern that can be used to identify them. If two patterns match, the samples are from the same person. So, the police are able to identify the man who burgled my house using his unique DNA fingerprint. They make a DNA fingerprint by taking a sample of cells from someone and separating fragments of DNA to make a pattern. If the pattern matches a sample from a crime scene, that person was there.
Okay, so how do you determine if that suspect is guilty or not guilty, and how do you analyze these fingerprints? As I mentioned, you can use these DNA profiles for a variety of reasons. If we're looking at trying to determine who's the daddy here, so we have DNA from Mary the mom, we have DNA from the child, we have DNA from Bob and Larry. You just match up these bands, and the child would have to have a band from every band the child has to match up with either Mary or one of these two. So if you look, you can see, well, there's a match with Larry, there's a match with Larry, there's a match with Larry. So Larry's the dad. And then when you look at crime scene, let's say we're trying to figure out, this is a real fingerprint, so you can actually see. One of the things I forgot to mention, let me back up for 1 minute. When you're viewing this, you can either look at it directly from the gel, as you see here, or you can use a blotting technique where you can blot it onto some paper and using radio or X-ray type mechanisms, as we saw with gene probes, and then you can get something that looks like this on paper. So this would be something you would want to take into court rather than the actual gel. So here you just look for some real obvious bands when you're trying to match these up, and you can see these first two bands, for instance, are only found here in suspect 2. And then if we just look at some of these other prominent bands, you can see, yes, suspect 2 is the one. That rules out 1 and 3 because they're missing these first two. So that's how you read those profiles. So another question that comes up is how do we sequence DNA? How did we know, for instance, in COVID-19, how was China able to send out the whole sequence so quickly? And the way we do this is through, again, an automated type of system that can read pieces of DNA. So relatively short DNA fragments can be sequenced by the dideoxy chain termination method. And it's an automated method where modified nucleotides called dideoxyribonucleotides, or DDNTP, attached to synthesized DNA strands of different lengths, and each one of these is tagged with a distinct fluorescently labeled marker that identifies the nucleotide at the end of each DNA fragment. They act as almost like roadblocks, so we know at the end of each piece, depending on which one we're using, because there's 4 different ones, A, T, G, and C, that we will know at the end of each of those fragments, there's either an A or T or G or C. So and then we can use a spectrogram using light, actually laser lights, to interpret it. So let's say we have this sequence right here that we do not, we want to identify this sequence. So we take the unknown piece of DNA and we put it in this machine. And of course, we have to have all the same ingredients needed to replicate any DNA, which means we need DNA polymerase, we need a primer, and we need a source of A's, T's, G's, and C's. And that's where these deoxyribonucleotides come in. But we want to fluorescently tag them, as you see here, so that anytime we see purple, we know there's an A, pink is going to be C, yellow is T, and G is going to be green. Now, you guys all know ATP, right? So we can use ATP for this, adenosine triphosphate, but this is cytosine triphosphate, thymine triphosphate, and guanine triphosphate. So again, if you look at each one of these, you can see that they are going to show up in a different color. So I'm going to show you this clip and then we'll finish it out.
To sequence DNA by the chain termination method, the DNA must first be obtained in single-stranded form. A mixture containing the single-stranded DNA, DNA polymerase, the 4 deoxyribonucleotides A, T, G, and C, and a single short primer is prepared. The primer has a nucleotide sequence that is complementary to the 3' end of the region to be copied, and is required so that DNA polymerase can initiate DNA replication. Equal amounts of this reaction mixture are placed in each of 4 tubes, and a different dideoxyribonucleotide is added to each tube. When a deoxyribonucleotide is inserted into the growing chain, replication continues. However, when a dideoxyribonucleotide is inserted, strand synthesis is terminated. The contents of the reaction tubes are then transferred to 4 lanes of an electrophoresis gel, and the oligonucleotides are separated by size and nucleotide type. The shortest oligonucleotide moves furthest down the gel. Reading from bottom to top, 1 base at a time, provides the correct DNA sequence.
Okay, so essentially what was happening there then is you take that strand, and remember each one of these is like a roadblock, and when you see that color, you know this is going to be a C, this is a G, this is an A. So you really have to start at the top here, work backwards by finding the complement. So I can tell you, I don't know this sequence, but I can tell you the complement is G, A, C, T, G, A, G, C. And sure enough, that matches up. But as I said, you can use this laser light and a detector to show that. And then you just get these printouts showing, again, everywhere there's a green peak, it's guanine, and then A would be adenine, C is cytosine, and T is thymine, as you see here. So if you look at something like the Human Genome Project, that even though they're trying to sequence the 6 billion letters in the human genome, it still was able to happen so much faster with the use of this kind of DNA sequencing mechanism. So much, much faster. Okay. So shifting gears a little bit here on page 120, concept 20.3. Looking at some things we can do with all this DNA technology. So I picked out a few things that we hear more about these days, but I wanted to start with just looking at some things that we've been doing for really quite some time. In fact, when I first got out of college, I worked at a tissue culture lab in Apopka, the indoor foliage capital of the world, where they were just starting to do tissue culture, where we would take fragments from our plants. We were actually growing ornamental plants, indoor plants, and we would take some cells from those plants and put them in a test tube with some culture medium, and it would actually grow a new plant. And so the reason that this was really nice, and this is nothing new, plants can reproduce asexually, no problem, but growing them in test tubes was great because you could grow so many more plants. You could start them this way, and you could transport them to different nurseries in these test tubes, and then the nurseries would finish growing them out. So it was a really big production, and the problem is, in order to do this, because you are dealing with media that can grow all kinds of other things, including different kinds of bacteria and fungus, you had to have a clean room. So that was rather expensive, and you had to dress up as though you were going into an operating room. You know, you had to, from your head to your toes, be covered so you didn't get any infections in that auger. Because if it got infected, say you got a fungus in the room, everything would be infected and trashed. So that's just typical cloning. We've been doing that from plant cuttings for centuries, but we can also do it in a tissue culture setting. But what about cloning mammals? In 1997, Scottish researchers announced the birth of Dolly. A lot of you probably remember or have heard of that. That was the first lamb that was cloned from an adult sheep by nuclear transplantation. So how did that happen? And unfortunately, Dolly did die in 2003. She didn't live very long. It was considered a premature death, and she had a lot of other issues. And so the speculation was that her cells were not as healthy as those of a normal sheep, possibly reflecting incomplete reprogramming of the original transplanted nucleus. So it was a huge breakthrough, and yet there's really not much to it when you look at how it's done. So you simply take cultured mammary cells. You can use any cells as long as they're diploid cells, and you have an egg from a donor cell, and you remove that haploid nucleus and take the diploid nucleus out of the mammary cells and put it into the egg. And so now it's almost like you had a fertilized egg, only the diploid cell nucleus is what formed it rather than an egg cell and sperm cell from two different parents. So the DNA is exactly like this mammary donor's cell. And so now the nucleus from that mammary cell is transplanted into a surrogate mother, and embryonic development continues, and voila, you get Dolly, the baby here, so which is genetically identical to that mammary cell donor. So this can be done in all kinds of animals. Since 1997, it's been done in mice, cats, cows, horses, mules, pigs, dogs. They showed this one Cece, the carbon copy was the first cloned cat. And again, but they don't always look or behave exactly the same way. And so there's some things that go on during embryonic development that seem to be affected by this cloning that we can't really exactly explain. So the big question always is, does human cloning exist? Well, here's some thoughts on that.
Since the birth of Dolly the sheep in 1996, scientists have managed to clone about 20 species, including cats, goats, and sheep. Now they've taken a step towards something a lot more controversial, cloning a human. What the scientists have done is created a human embryonic clone. They start by taking a human egg, take out the nucleus, and then they take a skin cell from a patient, take the skin cell and put it into this denucleated egg. The skin cell that's been transferred is sort of the software, and the egg acts like a hardware, and the hardware essentially boots up the software and creates an embryonic clone. From the embryonic clone, the scientists can derive other stem cells and go on to make heart tissue or nerve tissue or any other tissue that goes on to make a person. The goal of this research has nothing to do really with cloning. Cloning is just a tool. The idea is to create fresh tissue in a lab. If someone has a heart attack and you have scarring inside, you could use fresh tissue made in the lab to treat that scarring. If someone has a nerve ailment like Alzheimer's disease, you could transplant lab-made tissue into the person and hopefully treat that kind of malady. One of the biggest challenges in creating a human clone is that very few scientists are going to be willing to try this kind of experiment. The reason is it's incredibly controversial. It's banned in many, if not most countries, and it would provoke a huge outcry. However, there's always an outlier or two among the science fraternity who in some country, in some underground lab, may be willing to try and clone a human. Scientists have been able to clone various species, but they've made no headway really in cloning a monkey. And primates, like humans, seem to have egg cells that are a lot more fragile. So maybe that's one of the reasons why it's so difficult. For them to actually make headway in cloning a person, they'd have to refine the process a great deal more, and that seems to be a long way off, probably many, many years.
So there you have it. It is being done, of course, to make fresh tissue for medical use, but there's really no point in cloning people, at least it's not even ethically or even legal, really, in most countries. And there are problems, as I mentioned, associated with animal cloning, and as that video pointed out, there's many epigenetic changes not affecting DNA sequences. And epigenetic are things that we're just now learning about, things that influence the development of all species that we don't really fully understand yet. So there's a lot of problems associated with animal cloning, as he pointed out. So what about GMOs? I get asked a lot about GMOs because you hear so much about it, genetically modified food. Are you pro-GMO or no GMO? So a lot of people feel like anytime you manipulate anything genetically, you're creating monsters. And a lot of people are feeling like when this is done and people are told it's perfectly safe, that it's not. But this kind of genetic manipulation has been done for a long time, and so we use the same type of processes that I've been talking about to make recombinant DNA. So here's a good example of how we make a plant maybe that has resistance to pathogens or herbicides, where we can take a gene that has maybe, let's just say, pesticide resistance, or no, let's do the pathogen. So it's got resistance to pathogens, and we put that in, cut it out of the donor cell, and then combine it with the plant cell, and then simply introduce the plant cells into culture, as I mentioned before, grow those plant cells out, and now they have the new trait. The reason we might want to breed resistance to herbicides, for instance, is because that way they can go out and spray the whole field with herbicides, and then the target crop won't be affected. Or, so there's a lot of problems and thoughts about that because we don't want the use of so many herbicides in the first place getting on our food. So here's some pros and cons. This was the best video I could find that kind of gives you both sides of this story.
Are you tired of the media spinning the truth and pushing false narratives? Well, take a look at this. Over the last
2 million protesters in 52 countries rallied against Monsanto and genetically modified foods last week. So what's the dealio, and why are all these people taking to the streets? Hey everyone, you're watching D News, and I'm Lacey Green. About 90% of the soy, corn, sugar beets, cotton, and canola crops in the U.S. are genetically modified. Although it's not immediately obvious, most of the products in the grocery store contain GMOs, meaning that we've all consumed them. GMOs are plants that are genetically engineered by splicing their DNA with other species, producing plant, animal, bacterial, and viral gene combinations that you probably definitely wouldn't find in nature. This technology was developed because it produces a crop that resists pests and tolerates weed-killing herbicides. Now, GMOs also allow crops to grow in arid, dry land that they wouldn't otherwise. Basically, the major seller here is that it allows farmers to produce a lot more food in a lot more places with a lot less work. More supply means lower food prices, so without GMO foods, a trip to the grocery store could, in theory, cost more. How much more? I don't know. Probably a lot though, because they're a huge chunk of the supply. Also relevant, by 2050, the global population will hit 9 billion. If that doesn't sound like a buttload of people, just think about the fact that in 1950, the population was only 3 billion. Humans are like rapidly multiplying bacteria, ravaging the earth. All of those mouths to feed, coupled with the diminishing amounts of arable land, are a few of the reasons why some folks say, yay GMOs! But what about those protesters? 2 million people, 52 countries. The critics say that GMO foods come at a much higher cost than people are led to believe. There is research that suggests that GMO foods aren't harmful. This is why the FDA has approved them and say that they're safe. But there's also research that suggests that GMO foods are harmful. There are hundreds of studies that suggest that GMO foods and the herbicides that are on them make people more likely to get sick. Scientists at MIT and Cambridge have found that this effect occurs gradually, slowly eroding cellular systems throughout the body and triggering some of the most common health problems in the Western world. Gastrointestinal disorders, obesity, diabetes, heart disease, depression, Alzheimer's disease. Other studies have found that GMO corn contains dangerously high level of toxins and have linked GMOs with tumors and various types of cancer. I also find it a little fishy that all 41 of the studies funded by the biotech industry concluded in support of GMOs. Another concern of the critics is that GMO crops really can't be contained. Things like wind and water runoff are causing genetic trespassing into crops that aren't GMO. They're popping up in random places, and they've already contaminated up to 80% of organic crops in the Midwest. But there is talk of making another genetic modification that would make GMO offspring sterile. So this is merely the tip of the iceberg on GMO foods. If you're interested, I'd encourage you to do research of your own and see what you come up with. And let me know your thoughts down below. Are you pro-GMO or no GMO?
Okay, well, there's some good pros and cons of GMOs. I'll let you think about that. I'm going to go through a couple other types of technology that's out there. This is on page 121 of your course outline, looking at stem cells. A stem cell, as it says here, is a relatively unspecialized cell that can reproduce itself indefinitely and differentiate into specialized cells of one or more types. And stem cells isolated from early embryos at the blastocyst stage, which is an embryonic stage, are called embryonic stem cells, or ES cells for short. And these are able to differentiate into all cell types. The adult body also has stem cells which replace non-reproducing specialized cells, but they are not as prevalent. So there's a lot of debate about stem cell research because of the idea of collecting embryonic tissue in order to get these stem cells. So these stem cells are non-committed. That is, they haven't already decided whether they're going to be a neuron or a bone cell or a blood cell or whatever. And so that's why they're easier to manipulate. And so the fear was that with stem cell research that maybe women would start producing embryonic tissue and aborting it and selling it, and there's all these ethical considerations. So the idea was, well, let's stick with adult stem cells instead so we don't have that problem. So with embryonic stem cells, again, you can just take those embryonic cells, and you can actually manipulate that DNA in order to become those cells specifically, liver cells, nerve cells, whatever. Adult stem cells, on the other hand, are they're from your bone marrow, and they are designed only to form blood cells, whereas embryonic stem cells are designed to form all different kinds of cells, as I mentioned. So this brings us to this idea of human gene therapy. So gene therapy is an alteration of an afflicted individual's genes. And so gene therapy holds great potential for treating disorders traceable to a single defective gene. And researchers then had the idea, well, what if we can revert adult skin cells into embryonic stem cells? And they were able to do that, and they were able to produce what are called IPS cells, induced pluripotent cells. In other words, these are human adult cells that have been converted back. And those cells then can be used to treat some diseases and replace non-functional tissues. So the way that this works is, let's say you have a patient with damaged heart tissue or other disease, you remove those cells, some skin cells for instance, reprogram those skin cells so that they become these IPS cells, and then treat the IPS cells so that they differentiate into a specific cell type. And you can use a process called CRISPR to do this. This is gene editing, and this is really relatively new. Some of you may have heard of it, but if not, I'll show you a clip on this in a minute. Once you get these differentiated into that particular cell type, you return it back to the patient where they can repair that tissue damage. So this is this will work on what is known as regenerative medicine. The problem is this is all gene therapy, and when we say it's therapy, that means that those cells eventually die. So depending on what the problem is, you have to continue on with providing these genes, over the person's lifetime. So it is a therapy, it's not a cure. So this is a little bit about CRISPR. And as I said, this was a breakthrough in gene editing. And the way that this works, as you see here, the scientists can create a genetic sequence called a guide RNA that matches a piece of DNA they want to modify. And then it's actually the guide RNA along with a protein called Cas9 that acts as a pair of scissors will go in, hone in on the target DNA sequence, cut it out, and then put in the piece of DNA that we want in there. So it will actually do it on its own instead of you having to, take it out of the cell and manipulate it that way. So here's how this works.
Recognize that particular stretch of A's, T's, G's, and
Oopsie. Start that over.
in a document, if we suspect we've misspelled a word, we can use the find function to highlight the error and correct it or delete it. Within our DNA, that function is taken on by a system called CRISPR-Cas9. CRISPR is short for clustered, regularly interspaced short palindromic repeats. CRISPR consists of two components, the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA to be edited. To use CRISPR-Cas9, scientists first identify the sequence of the human genome that's causing a health problem. Then they create a specific guide RNA to recognize that particular stretch of A's, T's, G's, and C's in the DNA. The guide RNA is attached to the DNA cutting enzyme Cas9, and then this complex is introduced to the target cells. It locates the target letter sequence and cuts the DNA. At that point, scientists can then edit the existing genome by either modifying, deleting, or inserting new sequences. It effectively makes CRISPR-Cas9 a cut-and-paste tool for DNA editing. In the future, scientists hope to use CRISPR-Cas9 to develop critical advances in patient care or even cure lifelong inherited diseases.
So again, they're looking at CRISPR to find ways to fix all kinds of genetic problems. There's actually gels and creams that are being used for HPV, which is human papillomavirus, ear injections to correct the hearing loss. They've used it for skin grafts for insulin genes. They're trying to form drinkable or edible CRISPR probiotics to act as an antibiotic, and ex vivo therapy, that means outside, modifying outside the body for sickle cell and cancer. So here's a couple more clips on what's being done with CRISPR these days.
Here's the latest on xtalks.com. Researchers at St. Jude's Children's Research Hospital have pioneered the use of CRISPR gene editing as a therapeutic for sickle cell disease and other blood disorders. This is just the latest of many potential therapeutic applications of gene editing. Hello, I'm Sarah Massey, editor and webinar moderator for xtalks.com, and welcome to this edition of xtalks Vitals. While both fetal and adult forms of hemoglobin are responsible for carrying oxygen in red blood cells, they differ based on their molecular makeup of 4 subunits. Adult hemoglobin contains 2 beta subunits, whereas fetal hemoglobin consists of gamma subunits instead. As sickle cell disease and beta thalassemia are both caused by mutations in the gene encoding the beta subunit, the symptoms of disease slowly begin to manifest themselves as the adult hemoglobin levels increase. Mutations in the beta subunit can impair the oxygen carrying capacity of adult hemoglobin, leading to red blood cell death and hypoxia in vital organs and tissues. Since sickle cell mutations do not have an impact on fetal hemoglobin due to the lack of beta subunits, this form of hemoglobin has the potential to effectively carry oxygen in adults. By applying CRISPR gene editing, the researchers sought to prevent the gamma to beta switch of hemoglobin in adults, thereby reducing the symptoms associated with sickle cell disease and beta thalassemia. When applying the CRISPR technique to the DNA of stem cells collected from patients with sickle cell disease, they were successful in stimulating those cells to express healthy fetal hemoglobin. Subscribe to the xtalks YouTube channel by clicking the subscribe button below, and visit xtalks.com for more information on this story and others like it.
Okay, so treating sickle cell, and one other thing I wanted to share with you in working with cancer therapy. Now, this is not to be confused with the checkpoint inhibitors, the PD-1 blockers that I talked about when we were talking about mitosis. This is something different that involves more like designer drugs.
If it's working properly, the immune system wants to kill cancer cells. That's its job. Create an army of T cells to patrol our body for signs of infection and other diseases. The T cells inspect suspicious cells and, when they find an abnormal one, immediately lead an attack on it, while limiting the damage to healthy cells. The way they do that is pretty elegant. T cells have protein receptors on their surface, and these claw-like structures latch onto antigens, protein fragments found on the surface of all cells. When a receptor latches onto an abnormal antigen, like one found only on a diseased cell, the T cell turns on and releases toxic chemicals that damage the abnormal cell and recruit other immune helpers to the area. Sometimes cancer cells develop ways to fool the immune system, either by disguising themselves as healthy cells or by sprouting so many antigens on their surface that the T cells are stymied and can't mount an effective attack, or by finding ways to turn off the immune response against them. One innovative way to see through the deception is CAR T cell therapy. Thousands of a patient's own T cells are collected in a process similar to a blood donation. Using a modified and inactive virus that can't cause any disease but can introduce genetic information into a cell, the T cells are reprogrammed so they now produce special receptors called chimeric antigen receptors, or CARs, on their surface. These newly engineered cells, called CAR T cells, are grown in a lab, and millions of them are infused back into the patient. The new receptors enable them to latch onto a specific antigen on the patient's tumor cells and destroy them.
Okay, so this kind of therapy, again, it's therapy. You have to have this done continuously, but it does involve removing cells out of the body, engineering them using viral vectors, and then putting them back into the person. So I'm sure, don't know how expensive it would be, but I'm sure it would be because you're actually taking out blood and then, genetically engineering that and then reintroducing it. So lots and lots of different things going on with DNA technology. I could talk about this all day, but this video is the longest one I've made yet, so I'm going to stop here. And with that, I just want to tell you again, if you have any questions or comments, see me on the chat, email me. We will talk about this in one way or another. If you have any other interest, let me know. And the other thing is on the bottom of page 122, the General Biology Exam 5 review, the title should have been moved over to page 123. So if you're looking for that review, it is on page 123 because this wraps up the information for exam 5 now. So once again, hope you have a great day. Remember, for these long videos, there is a pause button, so sorry about that. But I hope everyone is well, and I'll see you next time or talk to you next time.